Okay, so the first time that I learned about the color blindness was the time that I was submitting a paper in a journal, and they told me that you can use the red and green in one figure because some people are blind to that. There is a concept comes back to the John Locke, the great English philosopher, that it's possible that two people use exact vocabulary for the colors, but they are systematically talking about different colors. Today we have Ariel that he's going to present an interesting experiment that we are learning more about this concept. Ariel is in the Monash University, and the title of his talk is Are Color Experience Equivalent Across the Visual Field? Thank you, Ariel, for joining us, and we are very looking forward to your talk.
Thanks, everyone. It's a little early here in Australia, so I've pre-recorded. If there's issues with the sound, then let me know and I can do it live, but otherwise I'll just, hopefully this works. Hi, everyone. My name's Ariel, and I'm going to talk about an ongoing project to determine whether color experiences are the same across the visual field. So my first guess at an answer to this would be obviously yes. My naive impression is that there's some external world. Light bounces off of stuff in it and into my eyes. This generates color experiences across my visual field, and I'm able to report these to you all. But there's evidence from neurophysiology and psychophysics to be suspicious of this naive view. Cone photoreceptor densities drop off dramatically with eccentricity, with most cones packed into just a couple of degrees of retinal eccentricity in the fovea. Correspondingly, visual acuity drops off rapidly with eccentricity. You start to get effects like crowding, where individual stimuli become harder to distinguish, and color contrast sensitivity diminishes quickly with eccentricity. So maybe the naive view isn't correct. How do we test this then? We need some way of capturing color experiences. One way to do this is to show people colored stimuli, which generate color experiences, and then get them to rate how similar these experiences are to each other. An intuitive take is that any particular color experience can be captured by its relationship to all the other color experiences. So to take red, for example, a red experience can be described as something that's identical to another red experience, very different from a green experience, and very similar to, but not identical to, a pink experience. Collectively, this set of similarity judgments describes color experience. Now, previous work has looked at these color similarity judgments in central vision, but to the best of my knowledge, no one's done this yet in peripheral vision. Researchers have looked at how people rate the same stimuli shown in central and peripheral vision along predefined axes, such as hue or saturation, but no one has collected actual similarity judgments from these peripheral stimuli. We want to examine similarity judgments from central to peripheral comparisons, and from peripheral to peripheral comparisons. Now, just before I get started on the experimental details, I want to flag that there's some controversy in the field as to whether we should be accounting for cortical magnification or not. Like with the photoreceptor densities, there's an uneven amount of cortical resources dedicated to visual processing across eccentricity, with much more being allocated for foveal vision than peripheral vision. Whether it's appropriate to scale the stimuli size to match cortical magnification is disputed, so I'll be showing examples of both cases. Now, to get into the experimental details themselves, we recruited our participants from Amazon Mechanical Turk. We did screen size and viewing distance calibrations initially to make sure participants saw stimuli all of the same size. In terms of eccentricity, the central stimuli were displayed at 1 degree of visual angle, and the peripheral stimuli were displayed at 10 degrees of visual angle. We used a range of different stimuli size where the peripherally scaled stimuli were shown using V2 magnification, which was 4.5 times the central stimuli at 10 degrees of visual angle. Obviously, for fixed stimuli, they were kept the same size. We used 9 different colors with 250 milliseconds of presentation to try and prevent saccading, and the whole thing took about 30 minutes. You can see an example here on the right where fixation, stimuli presentation, response. So what were the actual results? Now, very briefly, and I don't expect you to be able to interpret this, I'm just showing you the individual subject data to give you some idea of how varied and rich it is before I move on to the necessarily simplistic summaries. But you can see here there's quite a variety of individual responses. So to the actual data, you can see here that with cortically magnified stimuli, the color similarity judgments from central to central comparisons, central to peripheral comparisons, and peripheral to peripheral comparisons were all extremely highly correlated, suggesting that color experience structures are equivalent across the visual field. Looking again at this cortically magnified data, but now breaking it down by stimuli size, you can see that the central to central and peripheral to peripheral judgments remain highly correlated, regardless of stimuli size. However, looking at the fixed size stimuli data, you can see that below a certain size of stimulus, here below about 0.25 degrees of visual angle, these correlations start to break down. Now, this suggests that color experiences may not be equivalent when generated from sufficiently small stimuli. Now, one further question we had that comes from the philosophical thought experiment of color inversion is, how do I know that my red isn't equivalent to your green? This motivates the question of within subject color inversion, which is for the same stimulus and the same response, might the subject be having a different color experience? And if so, would we be able to detect this in the similarity structure data? So obviously we can't get at this directly because subjects wouldn't necessarily be able to notice it. So instead we simulated it and we saw whether we could detect the inversion. To do this, we take some set of stimuli and the similarity judgments that correspond to these stimuli. Then we swap the similarity judgments that apply to particular stimuli. So for example, here in the periphery, the judgments that apply to orange now apply to purple and vice versa. And you can see here what the similarity judgment matrix might look like once we re-index the judgments according to the original stimuli ordering. So what we want to know now is whether the central to central and peripheral to peripheral judgments will still be highly correlated with each other, or whether there'll be some statistically detectable decrease in correlation between the two apparent after we've switched which stimuli correspond to which similarity judgments. What we found is that in general, these inversions are detectable. You can see here on the left, one particular inversion that we simulated. So this resulted in a detectable decrease in center to periphery correlation across basically every subject, suggesting that if participants did indeed have this color experience inversion from central to peripheral vision, as described by that swapping you can see there on the color wheel, we would be able to detect it through a change in the similarity structure. So what do I want you all to take away from this talk? Firstly, that color experiences are probably equivalent across the visual field. Now, we've only looked here at up to 10 degrees of eccentricity, which is about the limit with what we can do with an online study on participants' own screens, but it's already well outside the fovea, as you can see on the bottom left there. After COVID, we'll try and follow this up in lab. Secondly, color experiences across the visual field when generated using sufficiently small stimuli aren't necessarily equivalent, but this isn't the case when taking into account cortical magnification. And lastly, although I only showed you one example, there's reason to believe that most within subject color inversions across eccentricity would be detectable. Now, all of this together is evidence that irrespective of veridicality, the naive impression of color experience across the visual field may be accurate, that the entire visual field is colored. Now, I'd just like to thank all of those who helped me with the work and to thank you for your attention. Additionally, if I could get you to really to fill out one really brief survey about your opinions about color experiences, I'd be extremely grateful. Okay, any questions?
Thanks.
Thank you, Ariel. That was a great study. I will wait a few moments to audience write the question. I'm pretty sure there are a bunch of questions right now. Yeah, so you also submit the document here. Do you want to add anything on that before audience start questioning?
No, I mean, people are, there's a question, there's already some questions.
Okay, awesome. Yes, yes, I see the questions. Okay, Julie asked that 10 degree may be outside of the fervor, but it's still considered in the peripheral regions. Do you expect the same result further in the periphery, like 20?
Yeah, so it's difficult to get at with the methods that we have at the moment, given we can't do in-laboratory studies. I would still expect the same results. I think if you do the cortical magnification and you use large enough stimuli, I'm suspicious you get the same results out. I don't know, but like considerably further. But that's really only based on my naive impression that my own color experiences extend pretty far out, rather than based on like good empirical data. Though if you look at the studies where they don't look at similarity ratings, they just look at how people rate the colors on like already predefined axes, you see that those are reasonably stable, I think, until a bit further out as well, with using, I think they're like 2 degree of visual angle stimuli or so. So my guess would be yes, but obviously I can't assert that without data.
Perfect, but I guess you have that in your experiments, the like up to 10, 20 degree, right?
Well, we have up to 10 degrees, which is
10
into
degrees.
peripheral vision, but it's not super duper peripheral, given humans can see up to about 200 degrees in total.
Awesome. A.M. Hound also asked, that's nice. Did you include RG diachromats as subject? Can distinguish the result from normal?
Yeah, so that's a good question. We didn't exclude anyone on the basis of saying that they had color blindness, but we did collect a post-experimental survey asking if they would report any color vision impairments or visual impairments or any of those sorts of things. So on my list of things to do is to go through those surveys, see if there's anyone who said that they were colorblind, and then see if they can see if their data looks different. But I don't have an answer for you yet.
How many subjects you had for that?
So each particular task has about 15 subjects in it, and I think there's like 10 different conditions. So there's about 150 subjects total. Yeah, it will be a good test to see if we can see the differences. Although it'd be interesting if I suppose you'd have to have compression along some axes, even if the relationships are still. Yeah, I agree. Sorry, I'm speaking to the chat. The comment was just that it would be a good test of the method if we can pull the colorblind people out.
Yeah. Oh, did you try on the colorblind people also? Yeah, that's what. Yeah. Okay, awesome. Thank you. Great. Also, if you want to come on the screen, just ask me to be on the screen for asking a question, Mario. Otherwise, thank you, Ariel, for your great talk. And let's move on to the last talk.